Just a quick review of passive transport. Looking at passive transport, we already said that it occurs when substances diffuse down their concentration gradient, like we have here in this diagram, where it's moving from the top of the hill down, and because it's rolling downhill, no energy is required, and particles are going to move until equilibrium is reached. The three types of passive transport that we've learned so far are diffusion, which is any molecule moving down its concentration gradient. Facilitated diffusion is any molecule moving down its concentration gradient with the help of a protein carrier or a channel. And osmosis is the diffusion of water specifically, so it's really just a special kind of diffusion. We're going to take a look at three different cells in three different solutions, and we're going to see which direction water moves across the membrane. The first cell we're going to look at here, the cell is the circle, and the beaker that it's in, or the environment that it's in, is the square. We have two solutes listed, and the solvent is water. Remember, we said water is the universal solvent. So here, the solute is sodium chloride. In the cell, the concentration is 5% sodium chloride. Outside of the cell, so in the beaker, the concentration is 5% sodium chloride. That means the concentration of the solvent, water, inside the cell is 95%, and in the beaker, the concentration of water is 95%. Now when we look at this, we know that sodium chloride can't cross the membrane. We've already said that things that are charged do not cross the membrane, and when sodium chloride is dissolved in water, it becomes the sodium ion and the chloride ion. It can't freely pass, but water can. Now we have to look at whether water is going to move across the membrane at all, and what kind of movement that's going to be. So we compare the concentration of the solvents. It's 95% water on the inside of the cell, 95% water on the outside. The system's already at equilibrium, so an equal number of particles move in and out, and we say that this is an isotonic system, where the concentration of the beaker is equal to that of the concentration of the cell. And we always have to specify whether we're talking about the beaker or the cell, but in this instance, they're both isotonic, iso meaning same. In this situation, inside of the cell is 10% sodium chloride, and the beaker is 5% sodium chloride. But again, we can't have sodium chloride move across the membrane, so we need to take a look at water. Inside the cell is 90% water. We're just, remember, adding up the concentration of sodium chloride and the concentration of water. It should equal 100. So 100 minus 10 is 90. Inside the beaker, it's 95% water. Water is going to be the thing that crosses the membrane here. It'll be osmosis. The beaker is higher in concentration of water than the cell. Because of that, water is going to move into the cell, and the cell is going to either swell if it's only a small amount of water that moves in, or it's going to burst if it's a large amount of water and it's an animal cell. It's not going to burst if it's a plant cell because it's aided by the cell wall. Instead, we just call that turgor pressure. In this instance, we label the beaker as a hypotonic environment. Hypo being less than. So this is a hypotonic environment to the cell. The cell here is hypertonic to the beaker, but we call this hypotonic. In this system, as we look at it, we have the solute concentration of the cell at 5% and of the beaker at 10%. The solvent, so our water here, inside the cell is 95%, and inside the beaker is 90%. In this instance, again, we compare our concentrations of water. We determine which is higher. It's higher inside the cell than it is the beaker, so it's going to move from where it's higher to where it's lower. So it moves out of the cell, and the cell is going to shrink. We call this beaker hypertonic. So the beaker is hypertonic to the cell of higher concentration. This movement of water across the cell membrane is called osmosis, but when we look at it in plant and animal cells, we call it plasmolysis. And that is when we examine whether the cell is going to shrink or swell or burst, and whether it causes pressure against the cell wall like it does in a plant, where it becomes turgid or it has a higher turgor pressure. This particular slide is just a summary of what we just looked at, where if we start in the middle at our isotonic solutions here, the cells are normal sized. Here on the left we have red blood cells, and on the right we have a plant cell with its chloroplasts distributed evenly throughout. And when you place that from an isotonic solution in its surrounding, and you decrease the solute concentration, then you end up having water rush in, which we had seen on the previous slides. Here we have some red blood cells that are swelling, some are bursting, like right there. In the plant cell, we see that the central vacuole fills up with water, pushing all of the chloroplasts to the edge of the cell. If we do the reverse process and increase the concentration of the solute, so it becomes a hypertonic solution, you'll notice all of the red blood cells shrink. And here in our plant cell, the plant cell itself shrinks with the cell membrane drawing away from the cell wall, but the cell wall remains in place. The last kind of transport that you guys are unfamiliar with is called active transport. And it's the opposite of passive transport because we're moving our object from the bottom of the hill to the top. So in other words, it moves from an area of low concentration to an area of high concentration. Just like every time in class we've mentioned sledding when we talk about passive transport, the fun part of sledding is going downhill. It doesn't require us to add any extra energy. The lame part that no one likes is having to move back uphill. That's the part that makes you tired. It requires energy. In this instance, the energy that it's going to require to move anything up its concentration gradient is called ATP, or adenosine triphosphate. If you've watched the sodium-potassium pump video already, you've seen the decomposition or breakdown of ATP driving the sodium-potassium pump. We use this for large, polar, or charged particles. The charged particles, again, it's like that sodium-potassium pump. Those videos are available for you linked on Plus Portals, but remember they are Flash player videos, so you have to be logged into a PC. You can't watch them from your iPads, but you need to watch the sodium-potassium pump video, the phagocytosis video, which is a type of active transport called endocytosis, and there's not an exocytosis video, but I have a secret. It's the exact opposite process of endocytosis. So if you just take that slider bar at the bottom of the video and play the video in reverse, then you've seen exocytosis. Endo and exocytosis are forms of active transport that involve moving things into the cell using transport vesicles. Phagocytosis is the taking in of solids, and pinocytosis is the taking in of liquids. Exocytosis is things exiting the cell using a transport vesicle. So it looks something like this, if you don't mind some rudimentary drawings. This is a cell, and it wants to take in this vesicle. So this vesicle is going to move closer to the cell membrane, where it's going to fuse its membrane to that of the cell. When it fuses, like that, find my eraser tool, then it's going to become part of the cell membrane and then release its contents into the cell. That process, when it's liquid, pinocytosis; when it's solid, phagocytosis. Exact opposite process is exocytosis, when the vesicle actually starts inside of the cell, moves closer, fuses, and then it disperses its contents out of the cell. That's everything you should need to know about active and passive transport. Don't forget to watch those McGraw-Hill videos. They're all available online. And if you're really thinking about studying, don't forget to do the questions at the bottom. The questions you are able to do on your iPad, but it's not necessarily the easiest. All right, I'll see you guys on Monday.